Good afternoon, all of you. Today we are going to discuss about gene family. Here we are dealing with definition and concept of gene family, their classification, simple multigene family, and complex multigene family, and developmentally controlled multigene family. So we can go through the topic.
Do you know what is gene family? Gene family is a set of genes within a genome that code for related or identical proteins or rRNAs. The members were derived by duplication of ancestral gene and then accumulation of changes in sequences. The members are related but not identical. Gene families are widely used in comparative genomics, molecular evolution and systematics. Here we take two DNA sequence of organism, and using computer analysis we check the similarities between the organisms. Through this we can identify the evolutionary history of that organisms. In more precise, we can say that a gene family is a set of several similar genes formed by duplication of a single original gene and generally with similar biochemical functions, which is a set of homologous or similar genes within one organism. When a single gene is present in two or more copies per genome, the condition is known as redundancy. The members of gene family exist as clustered together or dispersed on different chromosomes or present as combination of both. If genes of a gene family code for a protein, we can use the term protein family in analogous manner to gene family. One example for such family is the human hemoglobin subunits. Genes are categorized into families based on shared nucleotide or protein sequences. We know that when we take a functional genes which have a coding sequence called exons and non-coding sequence called introns. Genes are usually related only by their exons, with introns having diverged. Some members of gene family can evolve become pseudogenes. In a functional gene, in addition to introns, there is additional non-coding sequence called pseudogenes. Pseudogenes is a nucleotide sequence which cannot produce a functional gene product because of mutation. Formation of a multi-gene family occurs by three steps: duplication, translocation or relocation, and divergence. The initial event that allows the formation of related exons or genes is a duplication. Tandem duplication, which occur in the adjacent positions, which can arise through errors in replication or recombination. The duplication produces two similar copies of a genes. Separation of duplicates can occur by translocation, that transfers material from one chromosome to another. Members of a multigene family exist on different chromosomes due to relocation of these genes after duplication. Transposable elements play a role in the movement of genes. Duplication of intact genes, collection of exons, or even individual exons can occur. When an intact gene is involved, duplication generates two copies of a gene whose activities are initially indistinguishable, but then the accumulation of different substitution, the copies are diverged. The members of a structural gene family usually have related or even identical functions. although they might be expressed at different times or in different cell types. For example, different hemoglobin proteins are expressed in embryonic and adult red blood cells, whereas different actins are utilized in muscles and non-muscle cells. When genes have diverged significantly, or when only some exons are related, or their products can have different functions, the mutations allow duplicate genes to acquire new or different functions. Next is the gene cluster. Gene cluster is a part of gene family, which is a group of two or more genes found within an organism's DNA that encode for similar polypeptides or proteins, which collectively share a generalized function and are located in within a few thousand base pairs of each other. The size of gene cluster may change significantly, which is small, very few, or to several hundred genes. Genes found in gene cluster may observed near one another or in same chromosome or on different but which are homologous chromosomes. When gene product is needed usually in large amounts, extensive tandem duplication of a gene occurs. Example, gene for rRNA, gene for histone genes, etc. Sometimes all the members of a gene family are functional, but often some members are non-functional pseudogenes. Next is the classification of gene family. For understanding gene function and gene evolution, correct classification of genes into gene family is very important. Gene family classification is a grouping of genes or proteins into families, which yields important insight into gene evolution and gene function. When we examine or we cloned a new gene, to examine whether it belongs to a predefined gene family is the first task of a biologist. Leucine, the first protein database, was established in 1970s. To understand the functional significance, grand efforts have been made to classify proteins into families. As a result, a large number of gene families such as glutamate receptor family, ABC transporter family, and many gene families of the G protein coupled receptor superfamily have been curated. First is the multigene family. Gene family occurs as multiple copies of the same gene or multiple copies of related gene as a cluster, so it is called a multigene family. According to the structure, they may be either simple multigene family or complex multigene family. First is the simple multigene family. Here the same gene is repeatedly arranged on chromosome. Here all the genes of a gene family code for specific mRNA. Tandem repeats or array of rRNA genes code for rRNA genes, in which each copy is exactly or almost similar. E. coli genome contains 7 copies of rRNA genes. In human, multiple RNA genes present in haploid clustered on different chromosomes. Multiple copies of conserved rRNA genes on a given chromosome are located in tandemly repeated manner. Example for simple multigene family is the organization of rRNA genes in Xenopus. Do you hear about Xenopus? In rolling circle replication, we discussed about the rRNA of Xenopus. In genome of Xenopus laevis, 5 genes are scattered over all the chromosome, but remaining 3 are fewer copies and are restricted to single site called nucleolus organizer genes. In eukaryotes, 18S, 5.8S and 28S rDNA are seen as cluster, and 5S rRNA genes are also present in tandem repeats. DNA regions of chromosome is visualized at nucleolus, which forms expanded chromosomal loops with rDNA. In rolling circle application, we discussed that during oogenesis, the large number of rDNA is required for synapse. So, in order to produce large number of copies of genes responsible for the production of 5.8 is 18 is 28 is rRNA, because number of copies of genes are less with regard to this rRNA, and there is large number of fibers rDNA. Large number of fibers rRNA. So they perform rolling circle model. Next is the complex multigene family. It is the gene family in which related genes are repeatedly arranged, not as the simple multigene family. Here gene products are different. Example for such family is the organization of histone genes in sea urchin. Histone gene is the gene which codes for histone protein. Histone proteins are highly alkaline proteins found in eukaryotic chromosome. 4 different kinds of histone genes are recognized: H1, H2, H3 and H4. All these genes are organized as cluster, so it is called histone gene family. In sea urchin, histone genes are classified into 2 classes: early genes and late genes. Early genes are which express in the early stages of embryogenesis. They are present up to around approximately 300 copies per genome. Later genes are expressed later in embryogenesis development. They are present up to around approximately 10 copies per genome. Next is the developmentally controlled complex multigene family. Example is the globin gene family. A set of duplicated genes that encode proteins with similar but non-identical amino acid sequences called gene family. The encoded closely related homologous protein constitute a protein family. A few protein families such as protein kinases, transcription factors and vertebrate immunoglobulins include hundreds of members. Most protein family include from just a few to 30 members. Common examples are cytoskeletal protein, 7 kilodalton heat shock proteins, the myosin heavy chain, chicken ovalbumin, and the alpha and beta globin in vertebrates.
Genes encoding the globins are good example of gene family. We know there are 2 globin molecules, hemoglobin molecules and myoglobin molecules. Hemoglobin molecule is a tetramer which is composed of 2 similar polypeptides, that is alpha and beta genes. Genes are coordinately turned on and turned off during embryonic, fetal and adult stages of development. That is, there is difference in the presence of hemoglobin in embryonic, fetal and adult stages. Genes for alpha globin is situated in chromosome 16 and genes for beta globin is situated in chromosome 11. This is the picture for alpha globin genes, which has 4 functional genes and 1 pseudogenes. And this is the picture for beta globin genes, which has 5 functional genes, that is epsilon, G gamma, A gamma, delta and beta, and 1 pseudogene. All hemoglobins formed from different globins that carry oxygen in the blood, but they exhibit different properties that are suited to specific roles in human physiology. For example, hemoglobins containing either A gamma or G gamma polypeptides, which are expressed only during fetal life, because these fetal hemoglobins have higher affinity for oxygen than adult hemoglobins. So, they can effectively extract oxygen from maternal circulation in the placenta. Globin genes, which arose by duplication of ancestral gene, which is the result of unequal crossover during meiotic recombination in a developing germ cell. Germ cell means egg or sperm. Over evolutionary time, two copies of gene which resulted accumulation of random mutations. Beneficial mutation that confer changes in basic oxygen carrying function of hemoglobin and which were retained by natural selection. This result in sequence drift. Repeated gene duplications and subsequent sequence drift have generated contemporary globin genes. Two regions in the human hemoglobin gene cluster contain non-functional genes, which are called pseudogenes, which is similar to functional globin genes. Sequence analysis show these pseudogenes have same structure of exon intron as functional globin genes, which suggests these pseudogenes also arose by duplication of same ancestral gene. However, sequence drift during evolution generated sequences which either terminate translation or block mRNA processing. If they were transcribed into RNA, these regions become non-functional. Because such pseudogenes are not deleterious, which has no deleterious effect on human, they remain in the genome and mark the location of gene duplication. Other non-functional genes which arise by reverse transcription of mRNA into cDNA and integration of this intron-less DNA into chromosome. All globin genes are descended by a duplication and mutation from an ancestral gene which has 3 exons. The ancestral gene gave rise to myoglobin, leg hemoglobin and alpha beta globins. The alpha and beta globins genes which is separated in the period of early vertebrate evolution, after which this duplication generate individual clusters of separate alpha and beta like genes. When a gene has been inactivated by mutation, which accumulate further mutations and become pseudogenes, which means non-functional genes, which is homologous to functional gene but has no functional role. So this is all about gene family. I hope all of you understand this topic very well. Thank you.